Hello everyone, my name is Vahideh. I'm a postdoc in this faculty in the part 1 of experimental physics with Professor Madechik and Dr. Muravsky are working on development of IMS for different applications. Today, I'm gonna talk about detection of plant hormones by ion mobility spectrometry. Let me start by IMS, or ion mobility spectrometry. IMS is fast analytical technique for separation ions under electric field in an inert gas. Sample is heated to vaporize and enters to ionization region of IMS to be ionized by ionization source. Then ions are injected to drift tube by applying pulses to the shutter grid, and ions in a drift tube travel through the drift tube under electric field and reach to detector in different drift time. Smaller ions travel faster than larger ions and reach to detector in shorter time. Ions in IMS, ions are separated according to size, mass, and mobility. Each compound has its own peak that are used for qualitative and quantitative analysis. IMS has a wide range application in different fields. Since many years ago, IMS has used for detection of explosives, narcotics, illicit drugs at airports and other security checkpoints. In environments, IMS has used for detection of VOCs or volatile organic compounds in air. IMS is a powerful technique for in-breath analysis in medical and clinical applications. One important application of IMS is separation of isomers. Maybe you know isomers are compounds that have the same chemical formula but different arrangement of atom within molecule. Also, IMS is used for detection of pesticide in agriculture field, determination of additive and antibiotics in food chemistry, detection of medicine in pharmacy field, determination of trace amounts of chemicals in different matrix, and also IMS is used in gas and petrochemical, for example, for detection of ammonia in natural gas or a detection of sulfur compounds like H2S, CS2, COS, and mercaptan in natural gas. IMS has many advantages, including high sensitivity, fast response. It operates in atmospheric pressure, so it doesn't need to vacuum system, and this technique is non-expensive, and it's available from benchtop to portable and handheld design. This slide shows real photo of IMS and IMS coupled to mass spectrometry that are developed in our lab. One important part in IMS is ionization source. There are different ionization sources for IMS. For example, radioactive ion source, corona discharge, electrospray, photoionization, filament, and so on. We use corona discharge as an ion source in our IMS. Corona discharge is needle and front electrode that are connected to high voltage around 3 kilowatts, and it operates in positive and negative polarity. We developed a special design of corona discharge in our IMS. One of our projects is detection of plant hormones by ion mobility spectrometry. What is plant hormones? Plant hormones are a signal molecule produced by plant at extremely low concentration. Plant hormones play important role for defense of plant against different virus, bacteria, and other diseases. And also, plant hormones influence plant growth, seed germination, fruit maturation, and fruit ripening. One important volatile plant hormone is methyl salicylate. Methyl salicylate plays an important role in defense of plants against different viruses. We inoculated tomato plant by torus virus to induce production of methyl salicylate in plant. This movie shows how we inoculated tomato plant by torus virus. We designed and developed this setup for the determination of methyl salicylate in tomato plant. We use SPME or solid phase microextraction as a sampling method, and then sample is separated in MCC or multicapillary column and injected to IMS for ionization and detection. In this slide, I show one of the results of this project. Our results show that methyl salicylate content reaches to maximum level 48 hours after inoculation plant by virus. And also we observed upper leaf in tomato plant has a higher concentration of methyl salicylate. Maybe plant produce more methyl salicylate for defense of plants in new leaf. This work is already published, and our results show that SPME-MCC-IMS method is fast and sensitive compared to SPME-GCMS, and we need less sample weight, and total analysis time also is shorter than GCMS method. In another project, we detected VOCs emitted from blueberry leaves by Tenax-MCC-IMS. We designed and developed this setup for collecting the emitted VOC from blueberry leaves. Blueberry plant was placed in the glass chamber, and the emitted VOCs were collected by Tenax trap. It's Tenax trap. And after collecting sample, Tenax trap was transferred to heater for desorption, and then sample enters to MCC for separation, and finally enters to IMS for ionization and separation, for ionization and detection. We investigated three different stress to blueberry plants, including mechanical damage by punching, herbivore attack by aphids, and methyl jasmonate spray. In this slide, I show a response of blueberry plant to different stress factors. As you see, plant produce different VOCs with different intensity and concentration when exposure to different stress factors. Our results show that blueberry plants produce more linalool and benzyl acetonitrile after exposure to different stress factors. So, by identification of linalool and benzyl acetonitrile, we can find blueberry plant is under attack by some herbivore attack or like aphids, caterpillar, or other stress. In conclusion, we introduced new application of IMS in agriculture field, and we developed SPME-MCC-IMS method as a sensitive and fast method for detection of plant hormones. We developed Tenax-MCC-IMS for in vivo detection of VOC emitted from plant. In vivo means collecting of VOCs from living plants. And we hope Tenax-MCC-IMS method can be used further for inside measurement in gardens and greenhouses. I would like to thank Marie Curie Scholarship, Next Generation EU, APPV, and VEGA Grant Agency for supporting our project. And thank you for your attention.